Right, hi Year 12s. Thought I would do a screencast for this instead of a live. Just to make it a bit different, give you guys a bit of a break. So you should be getting this Wednesday morning. You can then get through the work at any point you want today. You're not tied to a specific time. We'll have opportunities to go through it the following week in lessons. So don't worry about if you don't think that you fully understand it. There'll be plenty of time for questions and making sure we get it. So we're not going to cover too much today, because as you'll see, the task, if you like, the work, isn't particularly challenging, but it's might take you some time. But I think it's important to do. One, it links back to something that one of you asked for earlier on, and two, if you can get these definitions correct, then I think it'll show that you have your understanding of the processes up to where we're at. So if you remember last time, we talked about macrophages. Okay, we discussed phagocytosis, and we talked about all of this creation of vesicles, formation of the phagosome, by fusing our lysosome vesicle with the vesicle containing the pathogen and its antigens. We now need to go, and we said that the cell, if it was a neutrophil or a macrophage, the cell could ingest multiple ones of these. It could then destroy itself and end up draining into the lymphatic system. Or there is an alternative scenario, which is what we're going to explore today, and it's going to lead us into the beginning of our understanding of the specific immune response. So up until now, we've been looking at our primary defenses, which are non-specific, our skin, our mucous membranes, our clotting, etc. Then we had our secondary defense, again, non-specific. This was our neutrophils and our monocytes or macrophages as they patrol the blood and the body. And if they come into contact with anything that they deem to be foreign, they will engulf it. And now we are beginning to look at our specific immune response. So how is this triggered? This is triggered by the creation of, down here you can see, this antigen presenting cell, or APC. And what can happen is that the macrophage, after ingestion and effectively digestion of the pathogen, the antigens on the surface of our pathogen can then become, as it says here, embedded in the macrophage plasma cell membrane on the outside of the cell. Once that happens, we refer to this macrophage as an antigen presenting cell, and it will circulate in the blood and effectively look to interact with T and B lymphocytes. And when that happens, when that interaction occurs, that is the start of our triggering of our specific immune response. So the specific immune response, what do we mean by that? It's where we end up with. It's the response that leads to production of memory cells, hence the term immune. It ends up leading to immunity for us, which means that we contain cells with the specific antigens to latch onto and antibodies to latch onto and destroy a specific pathogen. There are two pathways that make up our specific immune response. There is the cell-mediated response, and this involves our T lymphocytes, and our humoral response, which involves our B lymphocytes. So, if we look at our T lymphocytes first of all, these are all of our, as we saw, all of our leukocytes, our white blood cells, evolved from stem cells in the bone marrow. However, the T lymphocytes pass through the thymus gland, hence the T, where they are effectively, they differentiate a bit further and they're activated. Okay, they then circulate in our blood in very, very low numbers. And as you can see here, you don't need to know how, but it's really cool. We've got a whole load of genes which, when they're transcribed, can result in T cells with slightly different receptors, okay, on them. And like it says there, we can generate, we don't know how many, millions upon millions of T cells with slightly different receptors, okay, basically with the aim of being able to create a T cell with a receptor that is going to match the antigen on a pathogen. Okay, so how does this happen? The first thing is we get our pathogen invading our body. Then we have phagocytosis and creation of our APC. Our APC then circulates in our blood with the pathogen's antigens on the surface, okay, and they will eventually interact with a T cell with a complementary receptor, okay, to the pathogen antigen. Now, T cells could bump into an antigen and lock on and be activated that way as well. But as we have far more phagocytes in our blood, the normal way is for an APC to basically interact with a T cell with a complementary receptor to the antigen of the pathogen. This process is called clonal selection. You are selecting that clone, i.e. that version of the T cell, because there will be other T cells that have different shaped receptors that won't match the antigen, okay? And so you'll get an interaction with the APC and it will fail. And then another one, and eventually you'll find one that does fit. That's why it's kind of weird. Sometimes in a household, let's say, someone can get a cold. They can get, and then come down with it, and it can take them days and days, up to 2 weeks to really get over this. They have these symptoms for ages. They don't seem to get any better. And other people who are in the same environment maybe pick it up, maybe show some symptoms, but get over it very quickly. And this is one way to try and explain that, is that in some cases you're lucky and your APC bumps into a lymphocyte with a matching receptor and your specific immune response is triggered really quickly, and so you are not ill for very long. Other times you are unlucky and you have it until your APC finally bumps into a T lymphocyte or a lymphocyte and activates it, and then you get rid of it. And all that time you're still trying to fight off this infection. Okay, so that is clonal selection. Once that T cell is selected, it becomes activated effectively, and that triggers clonal expansion. So that T cell then starts dividing and multiplying via the cell cycle, mitosis, and it will also start to differentiate into 4 different types of T cell. Okay, you need to know the names of these, and you need to know what they do. So T killer cells actually attach to pathogens and destroy them. T helper cells release, move to the site of infection, but then release and cause other cell signaling molecules and chemical messengers to be released to stimulate and activate other T and B cells. T memory cells form, which will then remain in very low numbers, but they will remain in the body. Okay, for to effectively prevent reinfection. And then your T suppressor cells help to slow down and regulate the immune response, and slow down this clonal expansion once the infection is under control. So you need to know the 4 types of T cells and what they do. The T helper cells, we have to go into a bit more detail. So the T helper cells, here we said release some cell signaling molecules. These are called cytokines, okay, as the heading of the group of chemicals. So all 3 of these, monokines, interleukins, and interferon, are versions of, or are different types of a cytokine, okay? And again, you will need to learn these 3 and what they do in terms of how they help to effectively speed up getting over or resolving the infection. Okay, so that's our T cell, our cell-mediated specific immune response. What about our B lymphocytes? Well, our B lymphocytes, again, developed from stem cells in the bone marrow, but essentially, your B lymphocytes are going to produce, they're only going to produce two types of cell. They're going to produce the wonderful plasma cell, okay, and a memory cell, okay. And the plasma cell is going to be able to synthesize, create, and release millions of antibodies. Yeah, and we will be learning about antibodies next week. So when those antibodies are released, they then can again attach to pathogens because their receptor shape will be complementary, and they can then again slow down infection and help the rest of your T cells to destroy the invading pathogen. So how does this work? Again, our pathogen invades the body. Let's say we get phagocytosis, creation of an APC. That APC then bumps into a B cell or a T cell, and then a T helper cell attracts a B cell. Eventually, what needs to happen is that B cell with the complementary receptor, okay, to the antigen, to the APC, that B cell needs to be activated. Okay, again, clonal selection of the specific B cell with exactly the right shaped receptor. Once that B cell is activated, it then expands, okay, via clonal expansion and divides by mitosis. But this time, it only forms plasma cells, okay, which will go on and release millions of antibodies into the bloodstream, and memory cells, okay, which will remain in the blood just like T memory cells. So really, it's the plasma cells that you focus on with the humoral response. Okay, don't forget about B memory cells, but it's the plasma cells that actually are involved in helping with dealing with the infection. They release loads of antibodies. They can bind, like I said, bind to the antigen on the pathogen. Okay, and again, the presence of the antibodies helps stimulate phagocytosis, prevents the pathogen from reproducing, and does a whole other things. And we'll look at antibodies, like I said, next lesson. So, essentially, that's all I wanted to cover today. What I would then like you to do is, after you've watched this screencast, is to open up the other document here and start populating it with your understanding of and definitions of these key terms that we've been talking about. Now, I have included further up key terms from where we started with transmission. Okay, I've left out the pathogens, like the meningitis and, not meningitis, tuberculosis, HIV, athlete's foot, and everything, because you've basically filled out a table like that before, but you can combine them if you'd like. But here I have included the plant key terms, but just the ones that were new, so not cellulose, bark, things like that you would have come across, and really any of these key terms, like we had the clotting cascade there, and things like that. And really, by making you go and fill this out, I'm making you go back and reread and recheck your understanding of these terms, because as you said, there are a lot of terms, okay, coming in quite quickly. As you can see, we do have space for more. There are more terms coming. There probably, hopefully, aren't quite that many that I've put in there. But again, you can have this document and populate it for the rest of this defense and disease response unit until you've hopefully got a good summary. Okay, I appreciate that will maybe take some time, so you've got this double and any homework time or whatever until next week to get this done. But I do want to see that you've put in some detail. And again, I would encourage you not to just copy and paste. I would encourage you to type it out, write it in your own words, because again, that shows you that you have understood what the actual roles are. What are they doing? Yeah. Okay. Well, I hope you found that interesting, and I will see you next week. Have a good day.